It is my pleasure to introduce our speaker this morning. Biologist and herpetologist Dr. Tyrone Hayes is a professor of integrative biology at the University of California, Berkeley, where his research examines the role of steroid hormones in amphibian development, and he conducts both laboratory and field studies in the United States and Africa. Dr. Hayes is perhaps best known for his research on the herbicide Atrazine, where his findings reveal a crucial new link between conservation and health. He is featured in the PBS Nature documentary Thin Green Line, and in 2013, the Center for Biological Diversity presented its second annual E.O. Wilson Award for Outstanding Science in Biodiversity Conservation to Dr. Hayes for his work elevating the issues facing frogs and amphibian species worldwide. Thank you for coming, Dr. Hayes.
I want to thank you for coming out this morning. I want to thank you all for the invitation and for the opportunity to share a little piece of my life with you today. The piece that I'm going to share with you I called From Silent Spring to Silent Night, A Tale of Toads and Men. Yesterday I focused in the Save the Frogs conference, I focused on the impact of atrazine and other pesticides on amphibians. Today, in this Tale of Toads and Men, I'm going to talk more broadly about atrazine. I'll tell the frog story, but also talk more broadly about atrazine and its impacts on both environmental health and public health. Before I start, though, I want to share this with you. This is a phrase that I learned by working in Southern Africa I learned it from the Shona people, but loosely translated it means people are people through other people. And so before I ever begin a talk, I always thank and acknowledge the people that made me who I am. And among those, first and foremost, is my family. This is an old family portrait. At least they stopped taking them with me, maybe because I do that thing all the time. But among my brothers and my sisters, my parents, of course, important. I am a biologist, so as you know, I would not be here without Romeo and Susie Hayes, my parents. Wow, I've got on the same outfit. Anyway, that's a little embarrassing. I do have more than one, I assure you. But this is really to thank my wife, Catherine Kim, for her love and support throughout these last 27 years. Our 25th wedding anniversary is coming up. And finally, but my son and my daughter, Tyler and Cassina. And these are old pictures too. These are both prom pictures. And I'm not quite sure what made me more proud as a father, that my son borrowed my tie for his prom or that my daughter borrowed my earrings for her prom. They were both very important father moments. I want to thank all of the funders. It takes money to do work, and this is also my disclosure. The chemical industry, Novartis, Syngenta, has paid for my work, but they have since, as many of you know, decided they don't really like hanging out with me. That's okay. I'm still managing. I also want to thank all of the students that have been involved in the research, and important to me here, everybody in blue there is an undergraduate. I had a wonderful experience as an undergraduate myself, and I'm doing my best to try to give that back now that I have the opportunity to do so. These are my current lab members, so most of the newer work, I'll talk to you about some unpublished work, was done by them. Finally, I want to dedicate this to my grandmother who passed away in 2005. I was lucky enough, though, that before she passed on her love of education and her desire to make the world a better place through education onto me. Probably equally important, she taught me something, along with my father. She taught me how to tell a good story, I think. So I don't lecture, I don't give speeches, I don't give talks. I just want to tell you a story. And the story starts with a little boy who likes frogs. It'll end that way too, but one who's been affected over the last 47 years in some profound ways. This is a book that my mom sent to me when my son was born, and she wrote a note in there. I always start out with this book. She wrote a note in there that said, "This was your dad's favorite book." I don't remember the book, but I remember being in love with frogs for as long as I can remember. I've been trying to answer this question. It kind of all started here, what you're looking at there. This pointer doesn't show up here, so what you're looking at there is my grandmother's house. Her grandfather built that house. Plywood, tin roof house, no running water. My mom was born in that house. In that house, when my grandmother died, was still the papers and the bill of sale for when her grandfather bought her grandmother's freedom and married her. Still in that house. Probably equally important to me, though, is behind that house was a huge swamp, a huge forest. And I got lost in there as a child. We went down every weekend and stayed there. And I got lost for hours in this huge wilderness. And compliments of Google Earth, here it is. That's as big as it was. I don't know if you can see that. But some things haven't changed. The highways were always there. The cemetery was always there. And this area back here behind was a horse pasture. So that's as big as it was. But my memory as a child was that we'd get lost there for hours, and there were snakes, and there were lizards, and there were turtles, and there were these guys. And these, I see there's some kids in the audience, so I want to explain what's going on here. One of the cool things about frogs is they really help each other out. So you see that guy there, it's hurt his leg. This other frog's giving it a ride home, piggyback ride home. It's just one of the cool things that, and there's an inappropriate joke. She doesn't believe me. Even my undergraduates believe me, so you're smart. I could tell an inappropriate joke here about what happens when you help out a friend, but they lay eggs on the way home. And what you're looking at, and I'm sorry, you can't see the pointer here, but each one of the little spheres that you can see is a clump of wood frog eggs, about 2,000 eggs. And then, but hundreds in this species, hundreds of females will lay their eggs all in the same communal clump. And what's interesting about this arrangement, and this is, you'll see how this will lead to pesticides, but what's interesting about this arrangement is that the eggs in the middle, the eggs in the center can be as much as 10 degrees warmer than the eggs on the edge. And you have the same species here in Wisconsin. They're one of the first to breed along with the spring peepers. And what I became interested in this situation is, how does this behavior affect development? Because if you're warmer, you'll hatch sooner, you'll grow faster, you'll metamorphose faster, and I was interested in whether or not it'll affect whether or not you turn into a male or a female. And so what you're looking at here are the dissected testes. There's going to be a lot of gonads in my talk. Is dissected gonads. So these are, I don't know if you can see the little arrow there, but these little round things are testes. The bigger things on the one on your right are ovaries. And what I was also interested in is how the environment might alter whether or not you become a male or female, because that was known in reptiles and fish at the time. But I was also interested in the interaction with the genes. So what boundaries, for example, do sex chromosomes put on your ability to become male or female? Now, here's where we're at in my story. The reason the slides look so crappy is these are from 1989. This is from my honors thesis at Harvard. And these are, for the young people, for the students in the audience, this is before Adobe Photoshop. This is when cut and paste literally meant cut and paste. You had to cut the chromosomes out and paste them on the page, and that's where that function comes from in Adobe Photoshop in your Microsoft Office. Cut and paste. It had a profound impact on me. This was my first professional introduction into science. There I am at age 19. There's Bruce Waldman. And in fact, a good buddy of mine, Scott Valters, who works for DNR here, took this photograph. He was an undergraduate in the lab with me that summer. The other person there is Laura, who I'm guessing went into something else because she doesn't quite look as happy as I am to be in a swamp at 6 o'clock a.m. on a Sunday morning. But it had an incredible impact on me. Now, the other thing that I dreamed about as a kid was Africa. It was a dream. My father made $9,000 a year. First time I got on an airplane was when I flew up from South Carolina to Harvard. So going to Africa was truly out of the realm of possibility for me. But I remember folding out those National Geographic maps. You know, my father would bring home magazines. I remember watching the National Geographic show and Wild Kingdom and David Attenborough. And I remember dreaming of these guys, these National Geographic guys, exploring in Africa. Now that dream became a reality. By the time I was in graduate school, I started working in Advocacy, and it's a coastal forest on Kenya on the east coast of Africa. And National Geographic paid for it. I got to be in the magazine, I got to be on the show, I got to be in a Toyota commercial. I literally became that guy that I dreamed about. It was literally a dream come true. Now, it too had a profound impact on me. Because by working in the Advocacy, I discovered this frog where the males and the females look like they're not even the same species. And this is the kind of thing, the little boy who likes frogs, I'm just, wow, they're pretty. That was my first impact. So we brought these things home though, and see, now we had Adobe Photoshop. Look how neat that looks. Cut and paste, it's seamless. That's the same individual photograph once a day for 6 days, and it's about the size of your pinky nail. We discovered that they all start out green, they metamorphose green, and then the females at sexual maturity, months later, change color. So we hypothesized that estrogen was involved in this color change. So by analogy, in the same way that when females reach puberty, estrogen makes you grow breasts, we hypothesize when these guys reach puberty, estrogen makes them change color. And that analogy is going to be important for several reasons. And so we did some real simple, I do fancy stuff in the lab, but I do real simple things. Sometimes the answer is not go measure a gene and do PCR, but sometimes the answer is just simple. We just dip the frogs in hormones. And when we dip them in testosterone, nothing happened, but when we dipped them in estrogen, they changed color. So we tested and showed that our hypothesis was correct. Now here's where it gets weird, if it's not weird for you already. Here's the species Hyperoles argus. On February 15th, 1993, I'm giving this talk at UC Davis. I remember the date because it's the day before my son was born. My wife goes into labor. She starts having contractions while I'm giving the talk. So we're driving down the highway, heading back to Oakland where my son was born, and my wife, between contractions, says, "Oh, you should patent that." I thought it was the crazy hormones talking or something. I said, "You can't patent the frog. What are you talking about?" And so she called her brother-in-law, and he said, "Oh yeah, you can patent the frog. Here, 240, he's a lawyer, right?" So you get it. And so we did. We called it the Hyperoles argus endocrine screen, or the Hayes test. And here's where things really start to get weird. Here's why you patent the frog. The unexposed frogs are always green when they metamorphose. We can dip them in estradiol. That's the natural estrogen that's in everybody, if you're a female. Doesn't matter if you're a frog, a dog, a cat, a hog, or a human. If you're a female, this compound circulates in your body. If you give them ethinyl estradiol, a synthetic estrogen that's used in birth control pill, they'll change color. If you give them DES, a pharmaceutical estrogen that used to be given to women when they were pregnant, they'll change color. If you give them DDT, which is not a hormone, it's not an estrogen, but it binds the estrogen receptor and acts like an estrogen, they'll change color. So we screened dozens of compounds, I'm only showing you a few, and a student and I, Karen Menendez, showed that every compound that made my frog change color was also known to promote breast cancer. So we had a little frog that we could raise by the tens of thousands, and we could screen chemicals and test them for breast cancer activity. Not only that, you could send me some of your water, I could dip my frog in it. If it changed color, I'd be like, oh, he might want to drink that water. What's more is we showed that if you give these frogs estrogen and then give them a drug called tamoxifen, they won't change color. And tamoxifen is the anti-estrogen that stops breast cancer. So we could screen pharmaceuticals that then might go on for other screening and be used in the fight against breast cancer. That was the idea. So now a little boy who likes frogs gets introduced to some pretty big words. Intellectual property. The university says, yeah, that's a great idea. And either you show us how you're going to make money on it, or we're going to sell it to somebody else. So my wife, who had an MBA and an MPH at the time, and my brother-in-law, who was the lawyer, we set up this little company named after the forest where we collect it, Sokoke. And we got our first customer, Novartis, the biggest chemical company in the world. They said, we want you to look at atrazine. I'd never heard of atrazine. Now we're joined at the carbon bond. You Google Tyrone, you get atrazine. You Google atrazine, you get Tyrone. Here's what it looks like if you're into the chemistry. It's so-called S-chlorotriazine. It's an herbicide, so it's a weed killer. It's mostly used on corn. It's been used since 1958, so it's older than me. We use 80 million pounds a year in the United States. At the time, it was the #1 selling agrochemical in the world. It's used in more than 80 countries, but now it's outlawed in all of Europe. And now this slide is a lie. That's what the company lawyer tells me. They say atrazine has not been outlawed in all of Europe. It has been denied regulatory approval by the European Union. Now, I don't know what the difference is, but this one pisses them off. So this is the one I use, because that's just the kind of brother I am. The important thing is the company's based in Europe. So we're using 80 million pounds of a chemical that's not allowed in all of Europe, including Switzerland, which is not part of the European Union, but the chemical's not allowed. They asked us to look at another African frog, the African clawed frog. And they asked us to look at the African clawed frog because it's a more well-known laboratory model. Some people are shaking their heads. So you guys know about Xenopus? There were some here last night on display, the African. Yeah. Oh, it's illegal to do that in California. Anyway, but there's a funny reason why everybody uses this frog in science. Not because it's a great frog. It doesn't have a tongue, it can't jump, it's completely aquatic. It's not a great frog, but here's why everybody uses it. One of the other things that's unique about it is the human pregnancy hormone HCG will make this frog lay eggs. Somebody discovered in 1920. So literally in 1940, this was the pregnancy test. If you thought you were pregnant, you would go to the doctor, they'd inject some urine into the frog, and if the frog laid eggs, you were either happy or sad, depending on your situation. Now, I tell this story for a couple reasons, every time I tell this story. One is it shows the value of curiosity-based research, right? Who's the first guy who thought, hey, you know, I wonder what will happen if I inject pee into a frog? Like, how do you come up with that? Like, where does that idea even come from? I don't know. I've tried to find out. The second reason I tell this story is, just like my frog, the estrogens that make my frog change color are known to promote breast cancer, the human pregnancy hormone is so similar to this frog's hormones that it will make this frog lay eggs. This is a tale of toads and men because the hormones we use are exactly the same as the hormones these guys use. So as I tell you what atrazine does to frogs, you should be thinking, what might atrazine do to me? Because you're using the same hormones. And that's what we'll end up. The other reason I guess the story is significant is when they developed the other pregnancy tests, they threw these frogs out. And they still live in the environment. I was talking to somebody about invasives. So you can go to San Diego or San Francisco and collect African clawed frogs for a lot cheaper than ordering them through the mail. So I guess technically mine are African American clawed frogs, but that's a technicality. So we tested these frogs, and we found out that when you grow them in atrazine, their voice box, their larynx doesn't develop properly. Now that's bad news if you're making, you know, millions and billions of dollars on this compound, because male frogs sing and females don't because of testosterone. The same reason that men have deeper voices than women. I said that right, yeah. So these data suggested that these frogs were somehow affected in the gonadal region. You're going to see a lot of gonads today. So we dissected these animals that were exposed to atrazine, and many of them look like this. So that's an animal, those are testes, which is a good thing to have if you're a male. But then this guy's got ovaries, then it's got another testis, then it's got more ovaries. This frog could hurt its leg and give itself a ride home. There's a whole party going on right there. You don't even have to leave the house. And this is not normal. And when I say not normal, it's not a judgment call. When I say not normal, it's because I've been talking about these frogs for 15 years, and somebody always says, aren't frogs naturally hermaphroditic? And I finally asked about 10 years ago, and somebody said, oh, it's because in Jurassic Park, the frog DNA made the dinosaurs change sex. That's science fiction. There are no naturally hermaphroditic frogs. There are fish, as you may know, that are naturally hermaphroditic. There are no naturally hermaphroditic frogs. So this is not normal. So we made a hypothesis that that atrazine was an endocrine disruptor, which is what we were asked to test. So imagine this big yellow circle as a testis. Actually, imagine somebody you don't like, because we're going to do some bad stuff here. If you have a testis, you should make testosterone. A lot of people don't know this, but you know what the word testosterone means? It literally means testicular hormone. It's two words stuck together. So it's the male hormone. We hypothesized that atrazine turns on an enzyme, aromatase. I want you to remember aromatase, because we're going to talk about it a lot. Aromatase is an enzyme. It's the machinery, if you will, that converts testosterone into estrogen. The word estrogen literally means generator of estrus. It's the female hormone. So the hypothesis was that you're using up your testosterone. It's a little more complicated than that, but that makes the point. You're using up your testosterone, so things like your larynx doesn't grow, and other things that I'll show you. But also you're making estrogen, which is fine if you're a female, maybe not so much if you're a male. So you're feminized. You'll grow ovaries, and I'll show you some other things later. We could test this hypothesis by just measuring blood levels of hormones. So we measured testosterone, there you go, and control males. And here's atrazine-treated males, and here's females. So the atrazine was truly demasculinizing, or I like to use the word chemically castrating, because the company doesn't like that word, and well, you know, that's just the kind of brother I am. So here's where we're at in my story now. I published this paper. How many people know Proceedings in the National Academy of Sciences, PNAS? Big deal. I'm coming up for tenure. I'm, 30. I'm coming up for tenure. We published a paper in PNAS. I'm not saying the paper got me tenure, but it didn't hurt. I called my mom up. I call my mom every Sunday. I said, Mom, I have a paper coming out in PNAS. My mom drops the phone. I said, Mom, did you hear me? I have a paper coming out in PNAS. She said, yeah, I heard you, but how do you get a paper cut on your penis, son? I don't understand. And I said, this is a 100% true story. My mom's going to kill me. I said, no, Mom, PNH. She said, you ain't got to spell it. I used to change your diaper. My mom calls me up the next week. There's a point to this story. My mom calls me up the next week, and she says, son, I don't want to hurt your feelings, but how important is that magazine you were talking about? I said, what do you mean, Mom? She said, I went to Barnes & Noble. They never heard of that magazine. This is now My most important publication. It's a kid's book. I didn't even write it, but my mom can get it in Barnes & Noble. And I'll tell you why that's important at the end of this, that we do so much in the ivory tower that mean nothing to 99.9% of the world. And I've made a big change as a little boy who likes frogs. I've made a big change in how I do things because of that conversation with my mom. And we'll come back to that at the end. Now, as important as the paper, I should point out too, there were 4 black men and a Latina published this paper. It's probably a record for proceedings in the National Academy of Sciences. But as important as it was for all those reasons, there were some questions left unanswered. We didn't know if these hermaphrodites were males with ovaries or females with testes, which sounds like an easy question to answer, except remember one of the things I discovered back in 1989 is that frogs don't have visible sex chromosomes. We had a good idea, so if we treat with atrazine, we might get 50% females, 30% males, and 20% hermaphrodites. So we had a good idea it was the males affected, not the females. The other thing we didn't know is we didn't know what happens when they become adults. Again, we had a good idea, it sounds like an easy question to answer, but it takes these frogs 4 or 5 years to grow up. That means you got to get an undergraduate or even a graduate student and go, hey, I got a project idea for you, and maybe by the time you graduate, you might have interesting data. It was worse than that. It took us 8 years to figure this out. But I had tenure. I was in no hurry for me. Here's the answer. Another guy with a hurt leg. These frogs they're just accident prone. By the time the paper came out, there was a gene discovered by a colleague. I have a student in the lab in Japan now working there. There's a gene, you can see that top band on the gel, that only females have in Xenopus. So we could tell who was a genetic male and who was a genetic female. So for example, the guy who looks like he's smiling because he's getting a ride home, he's a male. That's his brother. So it turned out about 10% of the frogs completely turned into females. So there's one laying eggs, that's the same frog. If we didn't have the molecular material, if we didn't have the genes to measure, we'd think it was a female. In fact, this guy now, he's a grandmother. There's something you don't hear every day. He's a grandmother now. So they're completely transformed into females. Now, I could have published that, but I had tenure. I was in no hurry. I wanted to know what happens to the other 90% of the males that don't turn into females. Are they fully functional? I wanted to know, could they compete with other males reproductively? The problem is frogs breed in the spring, and so do undergraduates. Everybody wants to go away for spring break. Everybody wants to go away to some pool party thing or something like that. So I made a deal. I said, look, how about if you stay here for spring break, I'll give you a pool party, and we'll get another paper in PNAS. You guys would do that, right, if you were undergrads? It worked. It went something like this. It's a 100% true story. Here's what we did. Snoop Dogg wasn't there, it was a little more mellow, but here's what we did. We took 4 females, we took 4 control males, 4 atrazine-treated males, we just put them in the pool and see what happened. And we did this multiple times. And the idea, guys are thinking, that's not the sex ratio I want at the club, but the idea is the females are limiting. We want to know if these atrazine-treated males could compete. So we literally put them in there at 7 p.m. At 7 a.m. we come in and we just look at who hooked up. And you can probably can't see it from there, but there's surgical threads in the back so we could tell which ones are which. And so we literally just looked at how they competed. And when you do that, you find out that the atrazine-treated males almost never get the female. We only had 2 atrazine-treated males in 4 trials ever be successful. And then, I'm an endocrinologist, so I can't that's not enough for me. I gotta take some blood and measure something. So imagine you're at the club, the lights come on, somebody's writing down names, and they stick a needle in your heart and take a blood sample. It's not pretty, not a pretty party. On average, as you might guess, the testosterone levels are higher in the controls, the unexposed males, but more importantly, if you look at who made the love connections, the ones who get the females have much higher testosterone on average. And there seems to be that dashed line there seems to be some sort of cutoff that if you don't have testosterone levels above that limit, either the females don't like you or the other males just beat you up. And the atrazine-treated males just don't have enough testosterone to be in the game. Now, I probably could have published that, but I had tenure. I was in no hurry. I said, okay, they can't compete. Are they competent at all? So then we did a series. These are the pool party experiments. Then we did a series of something that I call the Motel 6 experiments. In this case, we just got them a room. We just put a male with a female, atrazine-treated male, female. There's no interference. You have the female, do your thing. The way we assay that is they lay eggs after their night of, and then we can see the one on the top left is unfertilized. All the rest are developing. So it was a real complicated test, right? There's an undergraduate sitting there going, 1, 2, 3, 4. almost quit. When you do that, you find out with controls, 85% of the eggs get fertilized on average. Atrazine-treated males, about 15. So they're impaired. And for two reasons. One is they don't even try. There's no competition. They sit there and they watch the females lay eggs. They don't even try, for the most part. Even if they try, you know what? I love this. Who likes p-values? You guys do statistics and stuff? I like a p-value, but you know what I like even better? When you can see that these are different. You can see that, right? That's a control testis under the microscope and a testis from an atrazine-treated animal sliced in under the microscope. And what you're looking at, if I blow this up, these are testicular tubules. I can't even see the pointer this close. But these are testicular tubules, and inside that dark stuff is all sperm. So just like we have got, or just like we hope we have. If you look at this guy, that's a testicular tubule that has a little cellular debris in it, but there's no sperm. So they don't have enough testosterone to do the behavior, and they don't have enough testosterone to maintain their sperm. So now we go back to PNAS, second paper. Atrazine induces complete feminization and chemical castration in male African claw frogs. See, I like that word because they don't like it. That's just the kind of brother I am. The other thing that's important here is that there's 7 undergraduates co-authored this paper and a Morehouse man. Very important to me. Now we're really getting into some stuff. I have a student, Mai Nguyen, and we set up these experiments with all males, 40 males, from control and atrazine treated from different families, because it turns out some families are more susceptible than others, and we're trying to understand why. And so there are 40 males. We know that they're males. They're all males genetically and morphologically. And Mai said, well, I have problems. It takes me a long time to clean out the tanks. And she was a brand new undergraduate at the time. She didn't know what was going on. She said, I have to pull them apart. I said, what do you mean you have to pull them apart? They're all male. And then she showed me that. And then she showed me that. I said, oh, we got some issues. So I didn't know what to do, and this stuff is unpublished yet. We're about to publish this summer. So I thought, maybe they've never seen females before. They've been raised with all males. Maybe, and they're all brothers, as in siblings. And said, maybe it's just big males overpowering little males. So that was easy to test. We went in one morning, and not the case. The ones in the bottom are always bigger, and females are usually bigger. What's more is the ones in the bottom are always the same. We put electronic pit tags in them so we can ID them. So it's not random. In fact, you can take the ones that are in the bottom out of the tub and the behavior goes away. So it's specific males that are mating with these other males. What's more is, because again, I'm an endocrinologist, if you measure their testosterone levels, the ones on the top almost always have higher testosterone. And the ones on the bottom, probably more important, have estrogen levels that are in the range of a female's. So they're males, but they're putting out a female hormone pattern and behaving like females. So they don't have enough estrogen to turn into a female. And see, we didn't test that before. That may be why they failed all the pool parties. They might have been actually behaving like females, we don't know. So that student now is going on to do her PhD, and she's looking at, I won't bother to explain what this is, but she's looking at areas in the brain that are sexually dimorphic to see if this early exposure to atrazine has actually feminized their brain, but not the rest of their bodies. She's also looking at their vocalizations to see whether or not they're actually singing like a female and actually attracting these males, and some of that might be the case. Now, the next thing that we did, and now I'm coming up for full professor, is we wanted to look at other frogs. We wanted to look at, because xenopus is weird, right? So we wanted to look at a North American frog, a common frog. So we looked at leopard frogs. And when we look at leopard frogs, we found animals that look like this. So that's a testis, and all that stuff in the bottom, I call it the junk in the trunk, all that stuff in the bottom, those are all eggs that have developed in this male's testis that are bursting through the surface of this male's testis. And there's an interesting thing about this story. At this point, I had a relationship, I guess that's what you'd call it, with the Environmental Protection. Anybody here from the Environmental Protection Agency? Oh, yeah. Okay. Oh, at this point, see, at this point, I had a relationship with the Environmental Protection Agency. And I remember I sent an email to Tom Steger. I said, look at this. And he wrote me back, I sent him all the data and everything. And he wrote me back and he said, well, Dr. Hayes, that's an interesting finding, but we're not sure it's one that would trigger reassessment and regulation of the chemical. My wife tells me there is nothing more painful in life than childbirth. And because I will never experience it, And because of the G.I. Joe Kung Fu grip she had on my hand when my son was born, I'm going to give her that. But fellas, I would guess that a dozen chicken eggs in my testicle would be somewhere in the top 5, wouldn't you? Tom Steger of the EPA says, no, that's not an adverse effect. Move on, spray on. I don't know, sorry. I don't mean to disrespect your agency, but anyway. So the next question we wanted to ask was, do effects occur in the wild? That was one of the main reasons that, that we looked at North American leopard frogs. Now to give you an idea, because I haven't told you this yet, we're using atrazine at 0.1 parts per billion. That's like 1/1000th of a grain of salt in 2 liters. I mean, if you do water chemistry stuff, I don't have to tell you that, but for me, I need a visual. To give you an idea of what's out there, the package of atrazine recommends application at 2.9 to 29 million parts per billion. So a farmer could be using this stuff at levels that are 290 million times higher than we use in the laboratory. If you go through the literature, these are the minimum and maximum levels detected in agricultural runoff, temporary pools, permanent water, and precipitation. Here's the levels we use in the lab. So here's all the levels that would be, all the areas that would be at risk. There's enough atrazine in rainwater to chemically castrate and feminize frogs. 500,000 pounds of atrazine come down into rainwater every year. It can travel over 1,000 miles. The USGS in Minnesota tell me that they can measure atrazine in the rainwater in Minnesota from applications in Kansas. What's more is, here's the EPA drinking water standard is 3 parts per billion. It's 30 times higher than we know to be biologically effective in frogs. So it's out there. We did a study. Well, we measured animals, we collected animals in the field, and I'll show you where we did that in a minute. I'm just going to show you some cross sections. If you're not used to this, just imagine I'm slicing up a salami. I'll fold out a slice, I'll blow it up, we'll blow up another slice. Those circles are eggs in the testis. We call them testicular oocytes. I made that word up, but now it's published. It upsets the company. But that's just the kind of brother I am. They said there's no such word. I said there is, it's published. Of course there's a word. Anyway, the point is, here's an old map from USGS of where most of the atrazine is used. Okay, Wisconsin, I think, is the only state that regulates atrazine, but there's still quite a bit of use there. We did a study where we collected animals, we controlled for latitude, and I'm being obnoxious. If you draw a line through those circles, that's Highway I-80, and we were going to a conference in Indiana, and we collected frogs along the way and got a nature paper. See, that's fuel efficiency, people, right there. Turns out that it's hard to find some place that doesn't have atrazine, but we found that every place we found atrazine, we found hermaphroditic frogs and vice versa. But the reason this has got into nature is that we could collect frogs from a blue dot where there's no atrazine and raise them in the lab in atrazine and get hermaphrodites. And we could collect animals from a red dot and raise them in clean water and get animals that develop normally. So we could show that it wasn't just natural variation or a coincidence. We could demonstrate cause and effect in addition to this correlative study. And then I became a full professor. Not just because of the paper, but it probably didn't hurt. The last little bit I'll tell you about frogs is that what we really wanted to do also is on a landscape like this one in Nebraska, we really wanted to ask how important is atrazine relative to other pesticides and also relevant to other environmental stressors. So for example, here's one example in York County, Nebraska. There's a runoff down there below that corn, but there are leopard frogs growing in that runoff. The problem is they don't just use atrazine, they use all those herbicides in red, all those fungicides in purple down there, and insecticides in blue, and that's just on one of those squares. Each square is using a different combination. The only thing they have in combination is that they almost all use either atrazine or glyphosate. But they're using a mixture. And so we did studies to ask how important is atrazine when all these other chemicals are being used in that runoff. We tested each one of these chemicals individually or in combination. That's what that looks like in my lab. It's done blindly, so the students, all they know to do is add the red, yellow, red solution to the red, yellow, red tank, for example. And then each of those frogs were raised, almost 3,000 individually in the deli cups you can see down there. And we monitored everything, growth, development, immune function. And I won't have time to tell you the story because I want to get to the of toads and men part. But essentially in my graduate work, I showed that stress hormones cause immune suppression, decreased growth, retarded development, and inhibited metamorphosis. We showed that pesticide mixtures, but not the individual pesticide, cause the same suite of effects, and we were able to show that essentially what pesticides do when they're mixed together is they activate the stress axis, and the more pesticides in your mixture, the higher your stress hormone levels, and then you get these adverse effects. We tested that further in Salinas, where we could do kind of a natural experiment. How many people know Salinas? How many people have eaten something from Salinas? Everybody raise your hand. 85% of the country's lettuce comes from Salinas. If you eat garlic, if you eat strawberries, if you've eaten greens, you've eaten something from Salinas, I promise you. 85%. So it turns out the river Salinas flows south to north, and most of the farms are in the north. Okay, and I'll show you what that looks like. So we had this really kind of natural experiment where we could look at frogs, and actually we're looking at an invasive. In Santa Margarita, here's where rich people get their water in Monterey. So that water's like 1.5 feet deep, 22 degrees, happy place to be a tadpole. Further down in Atascadero, however, it's not that much further, there's no pesticides yet, but all the water's being drained off for agriculture downstream. And so these frogs don't have pesticides, but you probably can't see that, but that's 3,000 tadpoles in a little pool of water that's about 1.5 inches deep, that's at 100 degrees Fahrenheit. So they don't have pesticides yet, but they're pretty stressed from environmental stress. Further down the water's back in Salinas, 1.5 feet deep, 22 degrees, but 100% of that water is agricultural runoff. 100% of that water is water that's coming off of your salad. Compliments of Google Earth. So we could go to Santa Margarita, pretty pristine place. We could go to Atascadero, high stress but no pesticides, and we can go in the same river, and that's about a 3 hours drive from one spot to the other. And in Salinas, no environmental stress, but high pesticides and fertilizers. I'm not going to show you all the data. This work is just being published this summer, but a picture's worth 1,000 words. These tadpoles are the same age, same species, same developmental stage, collected on the same day, just 2 hours apart. The only difference between this tadpole in Santa Margarita and this tadpole in Salinas is that the one in Salinas lives in water that's running off of your food. So again, here's a site. That arrow shows where we did all of the collections and some experiments that we did at Salinas. And now I want to take a sidebar, I don't know if that's the right word, to give a shout out to all those breath requirements that I had to take in college that I hated at the time, but now I appreciate it. Ms. Kaminsky would be proud. I'm going to read to you from John Steinbeck's East of Eden. It's written in 1952. Remember, Atrazine was introduced in 1958, along with a bunch of other pesticides. And here's what Steinbeck said about Salinas. Salinas was surrounded and penetrated with swamps with tube-filled ponds, and every pond spawned thousands of frogs. With the evening, the air was so full of their song that it was a kind of roaring silence. It was a veil, a background, and his sudden disappearance, as after a clap of thunder, was a shocking thing. It is possible for the night the frog song should have stopped. Everyone in Salinas would have awakened feeling that there was a great noise. In their millions, the frog songs seem to have a beat and a cadence, and perhaps it is the ear's function to do this, just as it is the eye's business to make stars twinkle. Here's what Salinas sounds like now. I haven't heard a single native frog call in the 7 years that we've been working on that river. And what I think is powerful, that wasn't a piece of scientific literature, but I think it's even more powerful because the frog song was so important to that landscape that a literary artist devoted a half a page to it. Not a piece of scientific literature, I think it's even better because that's gone now. That's our silent night now. And I'm not going to tell you that it's because of atrazine or that's because of pesticides. Many of you know 70% of amphibians are in decline. The main cause is habitat loss. I don't think anybody would argue with that. But in the middle of that, and this is a purposely obnoxious figure from a paper I published with my graduate students, is that pesticides are in the middle of that. When you lose the habitat because of agriculture, and the only water available now for you to breed in is water that's tainted with pesticides, you throw in invasive species, which are bringing in pathogens, put climate change on top of that, and you have pretty stressful animals with pesticides interacting with all of those four, what we call ultimate factors. This is a photograph from a place called Lake Nabugabo in Uganda. The great thing about working in Africa is you get to say the names like Arabuko-Sokoke and Lake Nabugabo. In Lake Nabugabo, the runoff from this crop goes in these containers as the sole source of cooking, drinking, bathing water for this village. And I show this because if I told the men in this village that the frogs in that water had the problems that I can show in our water in the US, it'd be clear. And there's no connection between environmental health and public health. There's no difference. It'd be clear here what environmental health means to public health. Whereas in my village, Not the Oakland A's or Raiders or somebody play. I don't watch sports. But I just live there in the hills, and my water, up at the top there, my water just comes from there. The difference is Uganda doesn't have a fancy EPA like we do, so we make assumptions that there's nothing bad in our water. Some of us make assumptions that there's no real connection between environmental health and public health. I don't think that's the case. I call this from silent spring to silent night because in much the same way that Rachel Carson thought that the death of birds and the role of pesticides in the silent spring was a warning to us, I believe that our little canary in the coal mine and its disappearance in our silent night is also a warning to us. A colleague of mine wrote in Ecoepidemiology, the occurrence of an association in more than one species and species population is very strong evidence for causation. Well, I've told you more than just correlative studies. I've talked to you about controlled experiments on more than one population, more than one species, more than one genera, but multiple families of frogs. What's more is, I'm going to tell you now, that other scientists around the world have shown similar effects of atrazine in fish, reptiles, birds, and mammals, including important studies that examine effects in humans. So every vertebrate class that's been examined. Now, I discovered this because I've published one paper already that I'll show you, but I have a big paper in the works where I emailed everybody in the world who works on atrazine, and I said, hey, let's publish one big paper. And we're doing that. The list is actually even bigger now. I did that in part because Syngenta started making personal attacks, right? Imagine you wake up one morning and this is the headline in Gawker magazine. Here's what the captions, and look at the keyword. How many of your professors could you search under that keyword? And then look at the caption here. I was like, oh. Oh yeah, could you imagine? My dean called me. Here's what inspired that. Revealed, here's another news article. How Syngenta investigated the press and shaped the news about its controversial weed killer atrazine. Another one, Syngenta paid third-party pundits spin the news on atrazine. And recently, now the whole story's come out. The New Yorker magazine just published a huge piece that's had a huge impact on my career and on my science. What they showed was that as a result of a lawsuit where Syngenta lost $105 million, they had to turn over all their private documents from their private meetings, which demonstrated something that I already knew, but now I can prove it. In hundreds of handwritten documents, look at their #1 goal under the science for dealing with atrazine. Discredit Hayes. Not like test the hypothesis, not like repeat the experiment, not like reanalyze this data. Discredit Hayes. They made a list. Here's a list I'm going to read to you. Look at this list of things that they were going to do to me. This is one of hundreds of pages. They made a list. Ask journals to retract. Investigate students. Investigate wife. School history. Family background. Consider suing for libel. Set a trap to entice him to sue. They made a list. They tried to buy my name on the internet. They, also on their list was exploit my father's health problems. And this is my favorite. They did a psychological profile. They held focus groups to see what to do to throw a black man off his game. And I knew this was going on, but they wrote it down. Everybody knows the first rule in the secret evil spy handbook is you don't write it down. And then if you do, you burn it and you shred it and you eat it and you self-destruct it. You don't write it down and turn it into a judge. And they paid somebody $10,000 to find out if I'm crazy or not. I'm not the one who wrote this stuff down. Who's crazy? Plus, you ain't got to pay nobody $10,000 to find out if Tyrone crazy. You can ask anybody who knows me and they'll be, yeah, brother, crazy. In addition, there ain't no good outcome. What's the outcome? They'll come back and say, no, he's not crazy. He just pissed off a black guy. But see, a lot of people get those two things confused. We're an emotional people. The other outcome is they say, yeah, he's crazy and he's whipping your butt. That's no good outcome. But anyway, I answered in a different way. I got 22 authors from 12 different countries, and we wrote a paper where I introduced the word gonadotoxin into the literature. They didn't like that when I make up words. But you know why I did it. Here's what we discovered. I didn't even know this. Here's my frogs I told you about. Right? On the right, on the left, control, right? You give them atrazine, no sperm in the testes. Here's a guy in Belgium, independently showed that if you give fish atrazine, sperm goes away. Here's a guy in Argentina. I didn't even know this guy. I know him now. He flew me down to Argentina. On the left is testes from a caiman. That's like a big alligator. You give it atrazine, there's no sperm, just like we see in frogs. Here's work that was done in both Croatia and Nigeria. I didn't even know the guy. I know him now. He flew me out to Croatia. But you take a rat, there's sperm in a testicular tube, you give it atrazine, there's no sperm. And then after the publication, I discovered this guy in Pakistan who's shown that if you take birds, if you take quail and give them atrazine, there's no sperm in the testis. So no matter what vertebrate class you look at, 22 scientists from 12 different countries around the world independently show the same thing. I might still be crazy. In fact, I'm proud of it. But they spent millions of dollars. And when they couldn't find anything wrong with my science, they launched a personal attack. That tells you about how bad their stuff is. So if you give atrazine, I told you, to frogs, testosterone goes down. That's why they don't have sperm. Is it a coincidence that this guy in England showed the same thing in salmon? Give them atrazine, no testosterone. Here's my frogs, here's rats. That work was actually done by the company. Every vertebrate animal we look at. Now, if you look at humans, we can't do experiments. Well, sort of. But here's a colleague of mine that's shown that men who have 0.1 parts per billion atrazine in their urine have low sperm count and can't get their wives pregnant. Now, that's just a correlation. But if you give a fish, an amphibian, a reptile, a bird, or a rat atrazine, their sperm goes away, and men who have atrazine in their urine, enough atrazine to chemically castrate a frog, can't get their wives pregnant in Columbia, Missouri because they have low sperm count. Now watch my PowerPoint trick. I spend a lot of time in hotels making these tricks. Because here's a study in California. That's field workers. Now watch my trick again. That's applicators. Men who apply atrazine have 2,400 parts per billion atrazine in their urine. Men who apply atrazine have 24,000 times the atrazine in their urine than we use to chemically castrate frogs in the lab. Men who apply atrazine have 24,000 times the atrazine in their urine than we know is associated with low sperm count in men in Columbia, Missouri. One of these guys could pee in a bucket. I could dilute their urine 24,000 times, and I could use the atrazine in their urine to chemically castrate 24,000 buckets of 30 tadpoles each. Now, what's their reproductive health like? We don't know, because they're primarily Mexican-American with life expectancies of 50. And in addition to atrazine, they're exposed to chemicals like chlorpicrin, which was originally developed as a nerve gas, 2,4-D, which was one of the components of Agent Orange. Now a little boy who likes frogs is introduced to terms like environmental justice, environmental racism. Because if you look at California, a few things you might not know. Somebody told me we're now 7th, but data suggests either 5th or 7th. If we were our own country, we'd be the 5th or 7th largest economy in the world. We'd be the 5th or 7th richest country in the world if we were our own country because of agriculture. 1 in 10 jobs are in ag, 30% of the land's in ag, 350 agricultural products, and here's one I didn't know. 50% of the U.S.'s food comes from California. 50% of the fruits, nuts, vegetables, and dairy come from California. Half of the U.S.'s food. We use more pesticides than any other state, and 90% of the workers are Hispanic. In red now are the top 10 counties for agriculture in California, about 30%. These are the counties that make us the 5th largest economy in the world. Where do you think the 30 poorest towns are in California? So the people who make us the 5th largest economy in the world are the targets of chemicals that we know are associated with adverse health outcomes. Environmental justice, environmental racism was a term that's new to me. Now it's one that I almost use on a daily basis. Does atrazine induce aromatase? I'm not going to talk to you about producing eggs. You're not going to grow eggs in your testes. But aromatase is important in both mammary cancer, breast cancer, and in prostate cancer. And their own factor in a community that's 80% African American, there's an 8.4-fold increase. They published this, an 8.4-fold increase in prostate cancer incidence, and that's men who work bagging the atrazine compared to men who work in the factory but don't work in the room where the atrazine dust is. There's at least one study in Kentucky that shows with a p-value, incredibly low p-value, that women whose well water is contaminated with atrazine are more likely to develop breast cancer compared to women who live in the community but don't drink their well water. And that's just a correlation. But if you look at rats, which are a proxy for us, atrazine causes testosterone to go down, like I already told you. Atrazine causes, though, a concomitant increase in estrogen, just like we've shown in fish, just like we've shown in birds, just like we've shown in reptiles, frogs. These are now mammals like us. And that increase, and this work was actually published by the company itself, that increase in estrogen is associated with an increase in the incidence of breast cancer in those exposed rats. It's just a correlation in humans, but we can show that these same types of effects occur experimentally in rodents. If you look at human cell lines, and I've published some of this work, and so is the company, if you take a human cell line that doesn't normally make aromatase and give it atrazine, it starts expressing aromatase and making estrogen. Just like we've shown in fish, just like we've shown in frogs, just like we've shown in reptiles, just like we've shown in rats, human cell lines do the same thing. I went to see him. I still think the company should be spelled with an I, not a Y, but it's just the kind of brother I am. They wouldn't let me in. They have a pipe that runs right into the Mississippi. 1.2 million pounds of atrazine flow into the Gulf of Mexico every year. 1.2 million pounds. Much of the community looks like this, and it's a community that's 80% African American. I mentioned that it's a community that's 80% African American for the following reason. These are the top 13 cancers you're going to get in the US. In red now, 11 of the 13 are the cancers that you're more likely to get if you're black, if you're African American. This is the mortality rate relative to Caucasian or white Americans. If you're black, you're more likely to die from 13 out of 13 cancers corrected for socioeconomic status and access to health care. And I can show you similar data for Hispanics. Here's my issue now. I'm a little boy who likes frogs, talking about environmental justice and wondering, does this reflect the biological difference between black and Hispanic people and white people, or is it that if you're black or Hispanic, you're more likely to live in and more likely to work in an area where you're exposed to chemicals that we know are associated with these adverse health outcomes? I'm all about combing for the cure, but how about combing for the cause? How about trying to stop us from getting this stuff in the first place? I think, to be cynical, there's just not money in that. And how about for all 13 of these cancers where there's racial differences in incidence and mortality, how about use cell lines from black or Hispanic people? Because right now there's zero. So most of the research that we're focusing on right now, if there are real biological differences or even exposure differences, may be irrelevant to the people who are more likely to get and more likely to die. Genetics may be irrelevant. When they tell you you're more likely to get breast cancer if your aunt or your sister or somebody in your family has breast cancer, is that genetics? Or is it because your aunt or your sister or your daughter are exposed to the same crap that you were exposed to? I'm just a little boy who likes frogs. One of my graduate students, and I try not to talk about my graduate students' work, showed that if you take a human breast cancer cell and expose it to atrazine, it'll start expressing aromatase. Just like we've shown in other cell lines, just like we've shown in rats, frogs, fish. That's important because of the following. Does it make any sense that most women get breast cancer after menopause, when the estrogen levels in your blood are lower than they've ever been in your life? It's because what's important is your lifetime exposure to estrogen and estrogen-like molecules. It's also because when you develop breast cancer, the cells around the cancer make their own estrogen. They express aromatase. That's why I asked you to remember it. And it's that local estrogen, even though it's low in your blood, that causes those cells, those damaged cells, to turn into tumors and spread. The local expression of aromatase is so important that right now the #1 treatment for breast cancer is a chemical called letrozole. It works by decreasing aromatase and decreasing estrogen so that your cancer cells don't grow and spread. Does that make any sense? When the #1 contaminant of drinking water does exactly the opposite, it turns on aromatase, increases estrogen, induces breast cancer in rats, and is associated with breast cancer in humans. It made sense to somebody, and here's the one they try to sue me over all the time. Novartis Oncology offers treatments for cancers that range from breast cancer. See, in the year 2000, the company that gave us 80 million pounds of atrazine, the exact same company was giving us an aromatase blocker to treat breast cancer. And by the year 2000, the same company that developed this new treatment for breast cancer to block aromatase was still selling 80 million pounds of a chemical that contaminates our drinking water that does exactly the opposite. I published a paper called The One-Stop Shop, Chemical Causes and Cures for Breast Cancer. They got mad. And they also said they wanted to correct me that their company is not in a black community because black people are there. It's in a black community because that's a low-income community and that's where black people happen to live. And I was like, you think that's better than what I said? In essence, I think that my interest in this aquatic organism above has told me something about an aquatic organism that when I went into this as a little boy who likes frogs, I didn't think about very much. We develop in water just like tadpoles. We're depending on the same hormones, testosterone, estrogen, thyroid hormone, just like tadpoles. And I would argue that one of my tadpoles trapped in a contaminated pond or a contaminated aquarium is no different than a fetus trapped in contaminated amniotic fluid. Studies now show that we're exposed to over 300 synthetic chemicals before we leave the womb. Most of them, we have no idea what they do. Atrazine causes prostate and mammary cancer in rats. We study rats because they're mammals like us. Atrazine causes immune failure in rats, which we've also shown in frogs. Atrazine causes neural damage in rats when they're exposed in utero. Atrazine causes abortion, and an EPA lab published this, when rats are exposed during pregnancy because it creates a hormone imbalance that won't maintain the pregnancy. Of those rats that don't abort, a second EPA laboratory published studies showing that the male pups are born with prostate disease. They're born with the prostate of an old man. Another EPA laboratory showed that of those rats that don't abort, the daughters are born with impaired mammary development, and when they grow up, their offspring have retarded growth and development because they can't make enough milk. It's this work that moved me in a very different way, not my own work. It's this work that made me realize that I'm a little more, I have to be a little more than a little boy who likes frogs. Because what struck me is that that rat on the bottom never saw atrazine. That rat on the bottom was affected by atrazine that its grandmother was exposed to. The rat on the bottom in this EPA peer-reviewed published study, that rat on the bottom never saw atrazine. That rat on the bottom was affected by atrazine that its grandmother was exposed to. So when I think about my little girl, she's 18 now, when I think about that my grandchildren could be affected by chemicals that we're using today, potentially my grandchildren's grandchildren could be affected by chemicals that we're using today, made me understand my responsibility is far beyond just a little boy who likes frogs. This is a study from a colleague of mine showing that in the dark area there, that women who get pregnant doing peak atrazine contamination, the line, are more likely to have birth defects when their babies are born. It's just a correlation. And I apologize for the young people. I'll show some images that you probably shouldn't look at. Here's another study that just came out in the last couple of years. I believe this one's from the Center for Disease Control. And what they showed here, and I'm going to read to you, maternal exposure to surface water atrazine is associated with fetal gastroenteritis. I didn't even know what that was. A student of mine now who's a medical doctor is working on this issue. Here's another study published in the last couple of years. And in this study, it showed that offspring of mothers with low levels of estimated residential atrazine exposure is associated with a twofold higher risk of choanal atresia. And finally, here's a study that just came out in the last year. And in this study, they showed, I won't go into details, it's entitled Case Control Study of Maternal Residential Atrazine Exposure and Male Genital Malformations. And I won't read it to you, but the study showed that there are 3 male genital malformations associated with exposure to atrazine during pregnancy. One of those is hypospadias. That's when the urethra doesn't come through the end of the penis. The other one is cryptorchidism. That's when the testicles don't descend into the scrotum. And the final one is microphallus, which is where the penis doesn't develop and grow. And what's interesting about these abnormalities is that we know that male genital development is testosterone dependent, is androgen dependent. And this is just a correlation, but in fish and amphibians and reptiles and birds and rats, we've shown that atrazine decreases testosterone and increases estrogen, and it's correlated with malformations that we know are associated with decreased testosterone and increased estrogen. My PhD advisor, I love him, but he had an attitude of many academics. He said, don't be an advocate, let the science speak for itself. I won't actually walk across and cross the line for you, but I believe very differently now. I believe differently because of statements like this from Syngenta. We assume no obligation to update forward-looking statements to reflect actual results. How could I let my science speak for itself? The other side isn't letting the science speak for itself. This one from the EPA disturbs me even more. The ultimate decision, they said about my work in 2006, is much bigger than science. The ultimate decision is much bigger than science. It weighs in public opinion. And when I first saw this, you know what I thought about? I thought about my mom. I thought about my PNAS paper. I thought about my Nature paper. I thought about the fact that the EPA is counting on my mom to have an opinion, for them to make a decision about her health. And here I am in the ivory tower publishing things that she has no access to, getting tenure and getting promoted for publishing in magazines that she can't get in Barnes & Noble. And that's when I realized I have a much bigger responsibility than a little boy who likes frogs getting tenure at a big university. There's a lot of problems. I love Obama. I voted for him twice, not in the same election. You may know this example where he made head of the FDA, a former Monsanto VP. The EPA's review of Atrazine, one of the members of the scientific advisory panel was paid by Syngenta and came back and presented to that same panel. The chair of the scientific advisory panel to the EPA was a Syngenta-funded guy when this all started. So there are big problems in the process. Who's ever seen this figure? The blue circles are the seeds that we use to grow our food. 90% of the seeds are owned by the red circles, the 6 big chemical companies. I don't necessarily have a scientific problem with GMO, but when the companies that own the seeds are pesticide companies, you can best believe that GMO is going to get us hooked more on the chemicals, not get us off the chemicals. There's a conflict of interest. I maintain a website. I've crossed the line, so to speak, so my mom can get access to my fancy papers. I helped Keith Ellison write a bill to ban atrazine. You can write to him and give him your support. He's in U.S. Congress from Minnesota. Groups that I've talked to have started a Facebook page. This is a group of students from Illinois, and I've helped write several petitions in Understanding the Science to try to get atrazine off the market. I've also been in a number of documentaries. This is one of my favorites, Last Call at the Oasis, where I got to play right alongside Aaron Brockovich. It was a big honor for me. I think that this is an old way of thinking, that we as academic scientists, we have to get involved. You might imagine I've been criticized because I've become an advocate or an activist. For example, here's a quote from Syngenta: Hayes, biologist turned activist. I'm no longer a biologist, I've turned. I was offended by this when they first published this, when they said this to the press. But you know, I'm a Harvard-educated brother. I didn't get upset at first. I went to my dictionary and I looked up activist, and it said, one that advocates or practices activism. I don't know about you, but I was always taught not to use the word in the definition, but this is straight out of Merriam-Webster here. So I looked up activism, and under activism it says a doctrine or practices that emphasizes vigorous action. And I immediately thought, that doesn't sound too bad. I don't mind having vigorous action. But just in case, I looked up vigorous, because I thought maybe there was some secret meaning I couldn't understand. When you look up vigorous, this is an adjective. It says possessing vigor, full of physical or mental strength, or active force. And I thought, I don't mind being an active force. What's wrong with that? But I looked up active force, just in case. And under active, it says characterized by action rather than contemplation or speculation. And I thought, wow, that kind of sounds like a scientist. And under force, it said a strength or energy, especially of an exceptional degree, active power. And I thought, that's me. I thought they were trying to insult me, and there they were giving me the biggest compliment that they possibly could have. I'm not a biologist turned activist. I'm a biologist, an advocate, and an activist. I feel proud saying that because somebody once before me said that those who have the privilege to know have the duty to act. Not only can you be a scientist and an activist, if you have the privilege to know, you have a duty to act. I trust the guy who said that. Thank you. I want to thank you again. I think we started a little late, but I think we have time for questions. I also want to say I understand from an anonymous tip that Syngenta has someone taking notes and transcribing everything I say, so I hope you got all that if you're out there.
If they'll share that with us, we'll put those on the webpage too.
I love it. Questions or comments?
Do you have time for questions? I have a microphone too to make sure that everyone in the back hears the questions. I'll start up front, but people can raise their hands and I'll try and work my way through the crowd. Dick?
So how long has atrazine been banned or whatever in Europe?
I believe it was about 2002, so I'm not going to take claim for it, but right about the time that my work and some other work started to come out, atrazine was denied regulatory approval by the European Union based on a precautionary principle approach that said if something's in the water that has the potential for harm, you have to prove that it's safe. Whereas in the US, you're innocent until proven guilty. It stays on the market unless the company proves that it's bad.
That's all of Europe. Is there one country, because I thought atrazine was banned like 20, 30 years ago in some individual country.
Yeah, so France banned it,
okay.
But they were still getting atrazine from the rainwater in Germany. So then it was banned by a couple other countries, so then the European Union said, well, given its transport and travel, my understanding is they made a decision for the entire union.
So that leads up to my real question. Are there any studies that are starting to compare the health impacts or the cancer rates in those countries that have banned atrazine for the longest period of time versus where we're at today in the U.S.?
You know, here's the problem. So the question is, can you look at cancer rates? It's the same problem as somebody asked me when I had birth control pill up there. Somebody asked, does birth control pill promote or reduce breast cancer? And every year it's a different story. The problem is it hasn't been around long enough for us to know. So if you're talking about something like atrazine, right? So exposure to atrazine in utero might lead to earlier breast cancer incidence as it does in rats. That means that if you're exposed in utero, you're more likely to get atrazine when you're 40, 50 years old. So it hasn't really been around or banned long enough. And in addition, in France, where it's been banned for over 15 years, last study I heard, the levels in the aquifers had not changed over that time period. So that's going to be a difficult thing to handle. And it's one of these, like, look how long we've been arguing about lung cancer and cigarette smoking. And that's a no-brainer. Now you're talking about something that you may have been exposed to in utero that you don't have evidence for that may have contributed to this condition down the road.
I live in Waushara County, which is just south of here, and I have atrazine in my water. I've had it tested, and I know it's there. And I spent a good deal of time last year trying to find a way to put a reverse osmosis system on my water, like we do for nitrates, to get rid of the atrazine. Very unsuccessfully. So I'm wondering if you know if there is a system that you can put in your home that's simple, not too expensive, that will actually get the atrazine out of your water.
Let me tell you why this question drives me crazy first, but thank you for your question. If 15 years ago I had made a deal with Brita, I'd probably be a rich man by now. So I get no money for saying this, but Brita Filter actually says on there, removes the contaminant atrazine. I don't know why it's on there, but it actually specifically, these filters specifically point out that they remove atrazine. So presumably it's removed by a filter. The reasons I say that are the people who are most at risk, those really high levels from factory workers and farm workers, that's by inhalation and absorption across the skin. So even if they had access to the information and the finances, they would still be at risk, and we still have the big environmental risk. But as far as I know, Brita and similar filters advertise that they remove atrazine. And by the way, we have done experiments in the lab where we built big charcoal filters and filtered water through. One of the experimental protocols, the idea is you put atrazine in the water, you get hermaphrodites, you can take that same water and take the atrazine out and not get the problem. So as far as I know, any of those carbon-based filters will work. The other thing to point out is the $105 million law settlement. That was drinking water districts suing the company for them to filter atrazine out of the water. So the other problem is it shouldn't really be our burden. They should really be, well, one, we shouldn't have it out there in the first place, but they should really be paying for the cleanup, not us as individuals. But thank you for your question.
Two quick questions. One, when you go and get a bottle of water, can you feel comfortable that that water doesn't have any atrazine in it?
Bottled water is a whole nother can of worms. There's not that I'm going to push my films, but you should see Last Call to Oasis orders a film called Flow, yes, For Love of Water, yeah. The film I was in that I can't remember the name of. A film called, you can get them both through Netflix or Blockbuster, but they go through all of the problems with bottled water. I mean, one, bottled water is actually not regulated. There's problems with water rights in some cases. Water, for example, one of the companies drains water off from under a Native American community because they're drawing off groundwater. In some cases, it's just bottled filtered tap water. And again, not that I'm knocking the bottled water industry, but somebody pointed out to me that Avion is naive spelled backwards. So but bottled water is not regulated. So there's a whole nother. So there's a whole other set of issues, and I would recommend either one of those films because it's not really my area of expertise. But both those films, Flow and Last Call, go through the whole issue of bottled water and issues associated with it.
So the answer is maybe. It might be in there.
I'd probably more likely recommend going with filtered tap water for my own personal use.
And that was the next question. If you get water from a municipality, do you think that they are testing for and removing atrazine from that water?
It depends on where you are. I know that my water, we get a thing maybe every 6 months or once a year, something like that, that lists the things they test for, and they do test for atrazine, and they tell you what the levels are, but it depends on where you live and what type of water you have in the company. I don't know that there's any universal law that says they have to test for, but they definitely don't necessarily remove. That's what this lawsuit was about. It was all about Syngenta paying the water companies to get the atrazine removed. So they definitely don't have to remove it, at least based on that. And that lawsuit was multiple states.
Got a question from way in the back here.
I can't see who's asking anything, so go ahead. All I see is a big bright light, so ask and I'll just talk out into the darkness, or to the lightness.
I'm from this county and do research on lakes and rivers, and the majority of the lakes and rivers that we've just done grab sampling in have detectable atrazine, but I'm wondering if you are familiar with sampling protocol that we could use to get a better handle on its presence in our local waterways.
No, so most of the work that we've done when we monitored atrazine, like in this we have multiple big studies where we've looked all over the country. I get that work done by the U.S. Geological Survey. There's a guy named William Battaglin. There is an ELISA kit that you can use. We used William Battaglin, who does some GCMS methodology because of because we know we're going to be criticized for whatever we do, so the samples are analyzed blindly by a government agency. So William Battaglin would have all the sampling methodology and analysis stuff, and so that's the place I would go for that. He's done a lot of atrazine work.
Can we get one quick, we got a follow-up on that one too real quick.
Sorry, it wasn't in terms of analysis, it's in terms of sampling protocol, so the frequency, the time of year, that sort of thing. We're in sandy soil, so we wouldn't be targeting runoff like you would in Illinois or somewhere with heavier soil.
I would recommend talking to William Battaglin. In our case, we just take a grab sample when we're taking the animals because we want to correlate. But he's done a lot of the data analyzing atrazine movement and contamination and transport and all that stuff. There's also a guy I believe named Perry Jones. I think he's out of Minnesota, also USGS. I remember him because there was a guy named Perry Jones who used to beat me up in high school. But it's a different Perry Jones. Yes.
Can you hear me?
Yes.
Do you feel safe with organic food?
Do I feel safe with organic food? It's always an awkward question because I say things that may not be relevant to most people, but I live in California, and I grow more food than I can eat. So almost all of my vegetables I grow, and there's no pesticides, there's nothing. I believe in organic food. Many places in the European Union have gone one step further. They do biodynamic, where they don't even use irrigation and water and all these kinds of things. But I never thought I would say things like this, but I do believe in organic and buying local, and when we do buy, we try to do those things. And organic means different things in different places, though. So I think it's more of a philosophy and more supporting a way and a methodology that's moving away from large-scale chemical farming. But organic doesn't necessarily mean pesticide-free. I'm sure you know that.
I see eggs at the store that say pesticide-free.
That's a different claim. That's one that's more protective, I guess, if your main concern is chemicals. Going organic also is making a lot of other political economic statements that I think if enough of us do it, we'll move in a direction that'll lead to a more less chemical use in the environment. I don't think we'll ever get away from, and I should point this out, I don't think we'll ever get away from using chemicals altogether, and I would never say that that's been my position necessarily. But in a case like atrazine, where we're using a chemical that has all these harmful effects that only increases corn yield by 1.2%, a crop that we eat less than 2% of as actual corn, to me that's a no-brainer. It shouldn't be on the market. There are other cases you can make arguments, and I'd be like, okay, yeah, we need to use some chemicals there. But I think in general, moving away from mass-scale chemical farming is something that we should. I remember when I was a kid, right, going away to the farm meant going away to a place that was natural and fresh and clean, and that's just not where we're at right now. You're probably better off, maybe better off in the cities, smelling the smog than going off to the farm.
Is there any legal active suit promoting or demanding that atrazine levels be eliminated?
Is there legal? Yes, there are legal cases locally. I was involved in some stuff in Tasmania. There was a big push. There was the one that was just settled for $105 million. I think there's another case coming up that I'm not at liberty to talk about. Hawaii is a mess. So in fact, my next trip here, or 2 trips from now, is 6 talks in 5 days on 5 islands in Hawaii. Most of the GMO and pesticide testing is done in Hawaii because they can do it year-round, and there's no right to know, so people have no idea. And here you're talking about an island that already has its own problems with ecosystems, with limited fresh water. You can go, for example, on Google Earth, you can go to Waimea, and you can actually see the silt and runoff in Waimea Bay from Google, running off from the chemical and seed companies' interactions there. So there's a big lawsuit in Hawaii. There's been a couple, mostly get settled, because these guys have lots of money. I think there was one in, I want to say Iowa, but
There's no national.
there's the bill that Keith Ellison keeps putting in to ban atrazine, but I don't know of a national legal. The $105 million settlement one involved multiple states. Illinois, I don't remember the other states that were involved, but other than that, there has been no big national movement. I don't know the answer to that. As I said, in France, the last time I saw somebody give a talk, it hadn't changed in an aquifer in over a decade. But it depends on, as probably somebody here already mentioned, depends on the type of soil, depends on how long it's been used, depends on whether or not it's surface water, how deep the lake is, whether it's in aquifers. If it's out on surface water with bacteria and UV light, my understanding, it can have a half-life of 8 days. But in other circumstances, that's not the case.
So atrazine is the example you worked on, but many other chemicals and pharmaceuticals where the research is really discontinued. And if you're thinking about monitoring water quality in your state, like for us right now, phosphorus, nutrients, that's been the focus. We're not prepared for measuring all of these other compounds out there. So if you just could comment on what you see as a way forward.
Geez, that's a good question. You're correct. There's a lot more out there than atrazine. As I always say, the best thing about atrazine is that we know what it does. Other stuff, not only is it not monitored as well as atrazine. The company likes to say atrazine is one of the most well-monitored chemicals in the world. Yeah, it is. So we know it's out there. That doesn't make me feel better. That okay, you can verify that it's in my drinking water. Oh, great. But at least we know where it is, right? We do know a lot about atrazine. For most of the other 80,000 chemicals out there, most we have. If you go on, and do a PubMed search of the scientific literature, you'll get lots of papers on atrazine, you'll get lots of papers on glyphosate or Roundup, you'll get some on metolachlor or maybe alachlor, and after that it just dwindles down to nothing, right? The great thing about atrazine is that we know a lot about it, we know that it's active, we know that it's persistent, we know that it's ubiquitous. It's the poster child for what we need to do differently. For these other chemicals, I have no idea. So if you're asking what's the next big hot one, I know metolachlor's out there. We've done some work with now S-metolachlor. There's a lot of concern for Roundup or for glyphosate. But again, the literature on those compounds is nothing compared to atrazine. The other issue is that it's not just pesticides. Somebody was just talking to me about pharmaceuticals and medical compounds used in medicine, BPA, or so there are other chemicals out there to be concerned with as well.
Back here.
Yes.
I got the impression that you may have an opinion on the EPA's structure. Do you see that we should be placing more science in the EPA? I believe the guy from whatever company that was, he's probably put there because of his management skills. Do we need management or do we need scientists in that? Do you have an opinion on how we could?
Let me say that for regional EPA and stuff, I've had good relationships. I've given talks with EPA, and I had some good relationships initially with the people in regulatory and EPA, with Tom Steger and Mary Frankenberry and some of those guys. But as you get higher up in the EPA, it gets very political in a way that I don't quite understand. Of course, the head of the EPA is appointed by the president. And that's one of the issues right there for atrazine, right? Where's the first presidential caucus? In Iowa. So you're not going to go to Iowa talking about regulating the number 1, number 2 chemical that's used on corn. And most of corn now, as you may know, is used for ethanol. So now it's all tied up in not just the farm bill and all those issues, but now it's tied up with the ethanol lobby, right? So it's a very difficult political question. Well, scientifically, it's a no-brainer. I think scientifically, the EPA, I think anybody who looks at the literature knows exactly what the issues are. The issues are, how do you battle the political and economic pushback on whether or not to regulate atrazine. One of the other difficult things that I also don't understand is that work that I showed you, those 3 studies in rats. There are multiple studies by the EPA. The EPA scientists aren't allowed to testify at the hearings. So I sat there, and I think it's because of conflict of interest, maybe because you can't have a scientist who works for the EPA testifying to regulate it. I don't know what the reason is, but I sat there and I watched Syngenta, I watched the company tear down those studies by the EPA while the author of those studies sat right there and just listened, and they couldn't say anything. The way that things work, like you can't raise your hand and say, wait a minute, that's not accurate. So it's a very strange structure. And also the scientific advisory panel does not make a decision, they make a recommendation. So the last scientific advisory panel on atrazine concluded that they only looked at 1 study. They eliminated everybody else's study. There's hundreds of studies out there showing atrazine's adverse. The only studies that say atrazine doesn't have any effect were all 100% conducted by the company on amphibians, right? So they looked at only 1 study, and guess who did the study? Not only was it a Syngenta guy, but it was a Syngenta paid guy that was on the EPA panel previously. So there are problems in there that I don't know how to solve. And I was very naive when I first went in, because I thought, oh, they'll look at the science and they'll say, okay, we can't use this chemical anymore. And then you saw, they're the ones who made that statement. The ultimate decision is much bigger than science. It's the same EPA in the New Yorker article that said, and I'm paraphrasing now, not quoting for who's ever taken notes for Syngenta's transcription. They actually said in the New Yorker article, you can go read it yourself, the EPA said they put a price tag on people's health. And they weighed that against the benefits of the chemical to the agribusiness. And my issue, again, it's a little boy who likes frogs who now has this environmental justice thing in his head, is you and I both know, if you think clearly about it, different people have different price tags on their heads. And if you're a Hispanic or African American or a low-income person, the price tag that they're putting on your head is different. The people who are most likely affected by the chemical has a different price tag that's being weighed against. The people who are applying the stuff and who are working in the fields have a different price tag than the people who own the farms and are selling the chemicals.
I have time for one more question. Right here.
You spoke earlier about the same company that was producing atrazine was also producing the medication that offset it. It's my understanding that we kind of do the same thing here. We banned DDT years ago, but it's still produced to be sold overseas by U.S. companies.
Yeah.
Is that correct?
So let me correct for the Syngenta note taker as well. So Novartis was the combination of AstraZeneca and Ciba-Geigy. Ciba-Geigy, by the way, made DDT a long time ago. So Ciba-Geigy and AstraZeneca combined to become Novartis. In the year 2000, just by coincidence, Novartis discovered that atrazine induced aromatase. They were the first to publish that. A guy named John Giese, who was the master's thesis advisor for the vice president of Novartis, discovered that. So in the year 2000, they discovered that atrazine induced aromatase. In the year 2000, they started selling letrozole, which blocked aromatase. And in the year 2000, the company split. Syngenta started selling atrazine, and Novartis continued selling the breast cancer treatment. So just by coincidence, in the year 2000, the two companies split up. But up until 2000, they were doing both. And you're correct, the DDT, and a lot of people don't understand this, DDT was never banned. Ruckelhaus, I think, was the head of the EPA then, did not ban DDT. Insects became resistant to DDT, individual states started banning DDT, and it became ineffective. It was never actually banned in the US. My understanding is for a period of time, it was regulated and could only be produced for military use and for export. So there was a period of time after 1972 that DDT was sold overseas, but my understanding now is that we no longer make or sell DDT. So I don't know if it was a matter of we were just selling the stocks that we had, but for some time, we'd sell DDT to Mexico, they'd spray it on strawberries and then sell strawberries back to the US. But my understanding is that's not true anymore, but that was true at some point. And it's still widely used, DDT, for malaria, fighting malaria and things like that.
And for people who are interested in sort of the DDT story, Wisconsin played a pretty pivotal part in its management and regulation. A local author from Stevens Point, Bill Berry, just had a book published by the university. It was actually the Wisconsin Historical Society Press. It was just published this spring in April. So if you go on to Amazon, you'll be able to find that book by Bill Berry is the author's name. I want to give Tyrone Hayes one more round of Lakes Conference appreciation.
Thank you.
And we're gonna send him home with a copy of Through the Looking Glass, our field guide to aquatic plants, something maybe he can just use to remember his trip to Wisconsin.
Thank you.